Welcome to this explainer. Today we're unpacking something absolutely wild, how a groundbreaking machine learning framework is finally bringing precision gut supplements closer to reality. It is this amazing intersection of synthetic biology and advanced computation, and honestly, it's going to completely change the way we think about nutrition. Let's get right into it. So, can artificial intelligence design the perfect human gut? I mean, it's a massive question, right? For years, assembling a stable microbial community from scratch to maximize health benefits has been this sort of holy grail. But now, a new framework is actually making it possible. To understand how they did it, though, we've got to zoom in on the incredibly complex microscopic world inside our digestive system. Section 1, the gut's math problem, the microscopic ecosystem. Inside all of us, trillions of gut bacteria are constantly cooperating to break down the food we eat. They're transforming complex dietary fibers into crucial molecules that literally shape our overall health. Before nutrients even reach our cells, these microbes break them down and ferment them, creating a diverse pool of molecules that dictate our risk for serious conditions, everything from obesity and diabetes to even depression. It's a bustling, highly active microscopic ecosystem in there. Now, one of the absolute most important molecules they produce is this hero metabolite called butyrate. Think of butyrate as the primary fuel source for your colon cells. It promotes anti-inflammatory signaling, and it heavily reinforces your gut integrity. When butyrate levels drop, well, that's when you start seeing links to inflammatory bowel disease, colorectal cancer risk, and metabolic disorders. But here's the catch. Producing it requires a delicate cross-feeding relay race. Primary degraders, like bacteroids or bifidobacterium, have to break down complex fibers into simpler substrates first. Only then can secondary fermenters step in to convert those substrates into butyrate. It's totally interdependent. And what makes this ecosystem so incredibly tricky to engineer is its extreme fragility. Adding or removing a single bacterial species or even just one type of fiber changes everything in totally unpredictable ways. As the researchers noted, each change creates a ripple effect that fundamentally and unpredictably alters ecosystem function. You literally can't just throw a bunch of random good bacteria together and expect them to produce butyrate efficiently. They interact, they compete, and they end up changing the whole environment. So here is where we hit a massive wall. The staggering mathematical scale of the problem. Get this. If you take just a modest panel of 15 bacterial species and 6 dietary fibers, combining them yields over 2 million possible conditions, over 2 million different ways these microbes and nutrients could interact. Relying on manual trial and error in a lab to find the absolute best combination? Completely impossible. Since human scientists can't test 2 million combinations by hand, they had to bring in some heavy computational lifting. Which brings us to Section 2, the AI optimization loop. Machine learning meets biology. To crack this, researchers utilized the Design, Trust, Learn cycle, or DTL. Basically, they treated the AI not as some magic wand that just instantly knows the answer, but as an iterative loop that gets significantly smarter with every single round. It's a strategy that's actually heavily used in protein engineering and material science, but until now, it had never really been applied to microbial community design like this. Here's exactly how it builds. First, in the design phase, a recurrent neural network predicts community composition, stability, and butyrate production based on initial inputs. Next, the test phase. This is where researchers actually roll up their sleeves and physically conduct experiments on the selected microbial combinations in the lab. Finally, the learn phase, where Bayesian optimization takes over. It looks at the data and selects the next set of experiments, prioritizing combinations that will either advance their health objectives or reveal how totally unexplored microbial configurations interact. Essentially, it learns from the real biology to make much better guesses next time. The result of this loop, honestly, extraordinary. They started with an initial data set of 387 combinations, and then let the model design successive batches of 288 new conditions. Over just 3 of these detail cycles, the model's predictive accuracy surged, with the Pearson R correlation jumping from 0.45 to 0.77. The algorithm progressively zoomed in on communities that hit the jackpot, meaning they were highly diverse, incredibly stable, and absolute powerhouses at producing butyrate. After grinding through all that data, the AI successfully pinpointed a champion formulation out of those millions of possibilities. Section 3, the winning combination. Finding the needle in the haystack. This brilliantly illustrates what the AI prioritized. It contrasted the winning formulation, which they dubbed HiCom plus IPX, against a low objective baseline known as LowCom plus XYL. The winning HiCom formulation consisted of a very specifically chosen 9-member bacterial community paired with a targeted blend of inulin, pectin, and xylin fibers. But here's the real question. Would this computational victory actually translate to messy, real-world biology? To prove this wasn't just some neat lab trick, they moved to ex vivo validation. They took this AI-designed champion combination and spiked it directly into complex fecal samples taken from 10 pediatric patients with obesity. The whole goal here was to see if this lab-designed community could actually bridge the gap and hold its own competing against a highly complex natural human gut microbiota. The results were staggering. The AI's chosen combination delivered a massive 5 to 10-fold increase in median butyrate production across the donors. And crucially, this is key, that 5 to 10-fold spike only happened when the engineered community and the specific fibers were delivered together, as opposed to giving just the bacteria or just the fiber alone. It completely, unequivocally validated the idea of perfectly paired symbiotics. Plus, not only did it pump out massive amounts of our hero molecule butyrate, but the engineered community was highly stable. It maintained a median relative abundance above 80%, even after 2 passages. Basically, this proved it could survive, thrive, and hold its ground alongside natural gut bacteria. This breakthrough right here lays the essential groundwork for a totally new era of precision nutrition. Section 4. The future of symbiotics. Beyond the gut. Very soon, we could be looking at a completely personalized vision of health, because this framework is entirely generalizable. Imagine this. First, we characterize your specific, unique microbiome. Then, we use AI to model exactly how different candidate symbiotic interventions would reshape your gut toward a desired state, maybe maximizing certain health-relevant metabolites or suppressing harmful ones. Finally, doctors deliver a perfectly personalized, optimized formulation made just for you. It's an incredible leap forward for human health. But I want to leave you with this provocative thought. If machine learning can actually solve the infinite complexity of the human gut ecosystem, what is stopping us from using this exact same design, test, learn approach to engineer complex microbial consortia elsewhere? Could we use it to heal depleted soil in agriculture, revolutionize biomanufacturing, or even engineer microbes to break down toxins and heal our planet? The possibilities are literally endless, and it all starts with understanding the hidden power of these microscopic ecosystems. Thanks so much for joining me for this explainer.